Hello and welcome to the Osmosis Egg Lab, where we will be learning about osmosis and selective permeability using eggs. So the first thing we're going to be needing is white vinegar. We're also going to need 3 large eggs. We're going to need syrup, and we're going to need salt and water mixture. In addition to that, we're going to need a jar container to fit 3 eggs, 3 individual cups for the 3 separate eggs. So before we get into the lab, we're going to need to define a few terms. The first being osmosis, which is a process by which molecules of a solvent tend to pass through a semi-permeable membrane from a less concentrated solution into a more concentrated one, thus equalizing the concentrations on each side of the membrane. This is going to be explored later on when we see the difference between the eggs in the brine solution versus the syrup solution. Another topic we need to cover is selective permeability. So a selectively permeable membrane is a membrane which allows certain substances to pass through and certain substances not to pass through. So the first thing you want to do when you start this lab is you want to get the 3 eggs and place them in a cup or jar filled with vinegar. These eggs need to be submerged in the vinegar for 48 hours. What this will do is the vinegar will actually dissolve the shell of the egg and leave behind the selectively permeable membrane, which can then be tested with the syrup and the brine. After those 48 hours are up, the next thing we're going to do is get 3 separate containers, one filled with syrup, one filled with vinegar, acting as the control, and one filled with brine or salt water. After these 48 hours with the vinegar, we're going to let those rest 24 hours in the new substances. After this 24-hour time period, we let the eggs rest in their new substances and perform osmosis and act on their selectively permeable membrane. The first experimental egg, which is the one placed in the syrup, gives us an example of how a selectively permeable membrane works. So the membrane is selectively permeable and does not let certain things pass through. In the case of the syrup, the sugar molecules could not pass through effectively due to them being too large for the membrane. This discrepancy allows very little substance to get inside the egg, and the egg ends up shriveling. The egg shriveling is caused by the need to maintain equilibrium. Since the solution outside the egg has a small concentration of water, because it's syrup, the water exits the egg, going into the substance. The egg ends up shriveling, and the syrup becomes slightly more watery than it was before it came into contact with the egg. The egg I ended up using shriveled a lot, but it ended up breaking. That's why you see a big dent in it. The next egg that we recorded was the control egg, which you can basically just see that the shell was dissolved and nothing really changed with the egg. And lastly, we have the egg that was placed in the brine. The second experimental egg, which was the one placed in the brine following the vinegar, gives us an example of osmosis. The water outside the egg is absorbed through the membrane, causing the egg to expand or to shrivel depending how much salt is in the brine. The equilibrium will actually move water into the egg or into the solution depending which has a higher salt concentration. In this case, the water I used was not as salty, so the brine had a lesser salt concentration, causing the water to move into the egg and the egg to expand. Since the inside of the egg had a lower concentration of water than the outside, the water moved into the egg. We can see with the egg that I used, it ended up becoming very expanded, looking like it's about to explode. It was also very squishy. So all in all, this egg lab is very interesting because the egg actually serves as a model of an animal cell. In an animal cell, there is no cell wall unlike the plant cell. So the outside of the cell is selectively permeable. It also kind of looks like an egg too. And that is the end of the lab. Thank you guys for listening, and I hope you learned as much as I did about osmosis and selectively permeable membranes.